Hello, hi, welcome everyone. My name is Julia Pollock, and I am the current artist and curator for the Art of Science program at the Carl R. Woese Institute for Genomic Biology. Our interdisciplinary science institute not only supports incredible genomic research, we also work to communicate science research to the public. Welcome. Will you introduce yourself and tell us a little bit about what kind of science you do?
Hi, Julia. Thanks for inviting me to this talk. So my name is Yanyu Xiong. I'm a third-year EC grad student from Professor Brian Cunningham's lab. So what we do is to using electromagnetics, optics, and nanostructure to develop new tools for disease diagnostic and life science research. We're also collaborating with Professor Sheng Wang and his student Neha to do some DNA origami work.
Cool, DNA origami. What is featured in the original images that you gave me?
Yeah, so the original images are showing a two-dimensional DNA origami, which self-assembled of synthetic DNA oligos with programmed sequence. So what we designed is a molecular pegboard or platform at a range of 80 nanometer times 80 nanometer that can arrange external elements with nanometer scale accuracy for different applications. And we using the Cypher AFM from IGB to scanning the sample, and I get an idea of how our structure forms and how the 3D structure look like.
That's wild. So wild to look at things on such a small scale. How does science imagery influence your research? Help you do your science?
In terms of the AFM calibration, so as you mentioned, those DNA origami are in nanometer scale, and we interested in their nanometer precision and how well they forms and how their structure look like. And basically the optical commercial microscope couldn't resolve since the diffraction limit of the light. So one can actually use TEM, the transmission electron microscope, to image it. But the TEM has a condition of high vacuum pressure, dry air environment. So those kind of harsh environment could somehow damage those little bit vulnerable DNA structures and make it explode or deforms. So what we found is best to characterize them is to using the AFM, the atomic force microscope, to use that atomic precision level of the tip to oscillating and tapping over the surface in order to get an idea of how their topological 3D structure look like.
Cool, that's really awesome. I really appreciated working with these images. This year's theme is called Elemental. So there is an earth piece and a water piece and an air piece, and your piece is the fire piece. I thought that DNA was sort of the fire of life, could kind of hold that space. So I used a evening night sky image to sort of pick up the light that was sort of in that light range. And then I was looking at a Swiss artist, Emma Kuntz, who did a lot of work with art and geometry and sort of graphing lines and wavelengths to think about healing through art and energy. And I really tried to think about what were the sort of wavelengths of fire or the wavelengths of DNA. So that's sort of how your piece is shaped.
Amazing. Thank you so much for your art design work. I actually feels like the fire pattern and the sunside view matches very well for the color of our AFM imaging, as well as the kind of scanning noise that we make in the AFM tip scanning process. So
that's awesome.
Thank you so much. Yeah, it's my honor to be working with you to create those science art.
Thank you as well. Have a good day.
Yeah, you too. Thank you.
Bye.